So let me just share my presentation. Can you see it?
Yeah, we can see that.
Yes, great. So thank you for the introduction, and thank you for inviting me, and thank you for showing up to listen to this talk. What I'll talk about today is, as Daniel said, I will talk about a method that we have developed for automatically quality controlling single cell and single nucleus RNA sequencing datasets. I put a link to a GitHub on here on the front page where you can go and find and install the package from if you want to. It's an R package that's that should be easy to install, and we are actively developing it. So just to make sure that we are all on the same page, I just brought a couple of slides to set the stage for why we chose to develop this method, and then after that, I will try to dive into the details and hopefully convince you that the method that we have developed is a good one for quality controlling your data. So since I was informed that the audience is a mixture of bioinformaticians or computer data science people and also clinicians, I just brought one slide about single cell RNA sequencing. By now, it's an older technology. Actually, it's has quite a few years. It has been existing for quite a few years now, but it's being really widely adopted now and being put into work in clinical settings and other settings. And what we had before single cell RNA sequencing was what is now called bulk RNA sequencing. And what we did back then, maybe 5-6 years ago, was that we took our tissue, a sample of interest, and we homogenized it and we purified the RNA and we did RNA sequencing. And that sort of gave us an average of the expression of genes in the cells that were originally in the tissue. And that, of course, masks any cellular heterogeneity that were in our sample originally. What a lot of people are doing now is to use single cell RNA sequencing or single nucleus RNA sequencing. And unlike the bulk methods, use isolate single cells or single nuclei. And from each of these cells and nuclei, you create a transcriptome, you sequence it, and that shows you the expression in single cells. And it also then unmasks this heterogeneity, cellular heterogeneity. And we can use this type of data for quite a number of things. First of all, we can identify cell types and states in our samples of interest. But we can also dive deeper into the biology and ask questions such as we can look for differential expression patterns. Here, for example, it's 2 cell types in 3 disease states where we can then compare them. It's also possible to do differential composition analysis, where we are asking if we visit this, let me just get a pointer here, where if we visit this again and we envision that this data was derived from healthy and diseased individuals, we could ask if there are, for example, more myeloid cells in the diseased individuals or not. We can also order cells according to their similarity and see how they develop in trajectory analysis. That can be a response to drugs. It can also be differentiation or general developmental processes. And along those lines, we can also use methods such as RNA velocity, which analyzes splice data to investigate or estimate future expression levels, and that can sort of give us a vector of direction for individual cells. And there are many other things that we can do with this type of data. But there are, of course, caveats and difficulties working with these types of data. And some of the first ones that we encounter is in sample processing. So we know for good reason that single cell RNA sequencing is limited to fresh samples. And we often do enzymatic digestion to release the single cells. And we also can see that processing delays can affect sample quality. And I will just show you a few pickouts from various papers to support these claims. So here, for example, Marsh and colleagues in a recent paper did a single-cell RNA sequencing on brain tissues, where they released the cells using either downsing or enzymatic digestion. And they found a very specific population over here of cells that is only present after enzymatic digestion. And they have a bunch of stress markers suggesting that cellular isolation can induce stress in these cells. In another study by Mazzoni, Bados, and colleagues, they purified the peripheral blood mononuclear cells, PBMCs, from individuals. And then they either processed them immediately, which is in gray, or they left them for several hours before cold preservation. And then they compared the transcriptomes. And here we can see that processing delays can significantly affect the quality of the libraries, as there is a significant shift in the distribution of the cells. So for example, within this dotted area is only T cells, and you can see that the gray and blue ones are on top of each other, which means that they're similar, whereas the purple, red, and yellow ones are moving away. And this can also be evaluated in principal component space, that there is significant shifts in the transcriptomes of cells depending on the processing. So one option is to use single-nucleus RNA sequencing instead, because that enables you to analyze frozen samples, and you can also process tissues that are hard to dissociate, and you can do that using mild detergents. You can also analyze tissues that contain cells that is not amenable to droplet-based single-cell RNA sequencing, such as, for example, adipocytes, which will float or be too big to be processed in regular microfluidics pipelines. However, even with single-nucleus RNA sequencing, we still need to be really mindful of how we are processing the sample. So the same authors here, Marsh et al., they also did single-nucleus RNA sequencing on brain tissues, where they either froze the tissue immediately or they left it for a few hours and then put it into the freezer or the cooling room and then processed it. And they can see here that even short-term storage introduces stress markers in these cells. So there's definitely also, even though we're using single-nucleus RNA sequencing, a necessity for processing samples rapidly. If we look at what we otherwise can find with single-nucleus RNA sequencing, for example, if we look at composition, Nico and colleagues have made a really nice study where they compare single-nucleus and single-cell RNA sequencing, and how they're able to recover the true composition of tissues. Here they have a truth data set from Clark et al., and then they compare single cell and single nucleus. And you can see here that, for example, if we look at podocytes, single cell is much better at recovering the true distribution, whereas if we look at the loop of Henle, it's actually the opposite. So I would say that single nucleus and single cell are both struggling a little bit to find the underlying composition, and they have both biases, but they do have different biases, which means that we shouldn't be comparing compositions across these types of data. What is the same, however, is largely marker genes and gene expression patterns. So here, for example, Santiago and colleagues have done a single cell and single nucleus RNA-seq on the retina, and they find that the marker genes along the diagonal here for these different cell types are largely the same across these two data modalities. So we can use single nuclei RNA sequencing to find cells, but we just need to be aware that we have a different bias towards composition changes. So what is then the downside to using single nucleus RNA sequencing? One of the downsides is that cell types can be harder to separate. Even though the marker genes are the same, we often see that the separation of cell types is less clear. Here, for example, a study by Gate, I will struggle to pronounce that name correctly, so I will not chop it up. They did single cell and single nucleus RNA sequencing on the same tissues, and what they find is that the cells in the single nucleus RNA-seq dataset are much closer to each other in these reduced dimensional spaces. Although UMAP space is not the best space to interpret this distance in, I think this represents what we are oftentimes seeing. The same can be seen in another study by Slüber et al., where they also did single cell and single nucleus RNA sequencing on the same cancer cells and find that cellular separation is less pronounced or less strong in single nucleus RNA sequencing. So why is that? And one of the reasons is conceivably shown in this graph here, which is taken from a paper by Fadl et al., where it's also single cell and single nucleus RNA sequencing on retina. And what they see is this is rhodopsin expression on the y-axis and different cell types on the x-axis. And they see that the rods in the single cell data express high amounts of rhodopsin as expected. We also see the same in the single nucleus RNA sequencing data, that rods are the cells that express the most rhodopsin, but there's actually quite high expression levels in all other cells. And this alludes to one of the issues with single cell one of the issues with single nucleus RNA sequencing, which has to do with ambient RNA. Ambient RNA are cell-free RNA molecules that are in our solutions when we purify cells or nuclei. It's depicted in this tube here, where these round things either represent cells and nuclei, and the swirly things represent free-floating RNA molecules. And in single-cell RNA sequencing, the problem is limited. Some of the sources for cell-free ambient RNA is leaky cell membranes and cell lysis, which happens inadvertently, but it's a limited problem that often gives rise to low amounts of ambient RNA. However, in single nucleus RNA sequencing, there's leaky nuclear membranes, but there's also a very intentful cell lysis where we release all of the cytoplasmic RNAs into solution. And this can be hard to get rid of and wash away. And this can often lead to higher amounts of ambient RNA. And in fact, this is supported by studies in the field. For example, here by Jansen et al., who did a single nucleus RNA sequencing, the last two graphs here, and single cell RNA sequencing in a setup where they can detect background RNA. And they find that single nucleus in both cases, although variable, is associated with much higher levels of ambient RNA. So if we imagine that we will do a single cell RNA sequencing or single nucleus RNA sequencing on such a sample like this, and we will do it using a fluidics-based approach like 10x genomics, we can conceivably find different types of droplets. We can find droplets that do not contain a cell, droplets that contain one cell, or droplets that contain multiple cells. And all of these stages will also at the same time be with various degrees of contamination from free-floating RNAs. And this leaves us with at least 3 computational problems when we look at the data initially. The first problem is how we can distinguish between contaminated empty droplets and cell or nuclei containing droplets. Second question is how we can distinguish between singlets and multiplets. And the third question is how we can distinguish between contamination and signal within droplets that contain only one cell. And today, what we will talk, or what I will talk about, is only the first question, which is how we can distinguish between contaminated empty droplets and cell or nuclei containing droplets. Sorry. There are already several tools out there for this task, namely these 6, at least that we could find scouring the literature. And 4 of them are published and 2 of them are preprints currently. And then there's ours as well, which is of course also a preprint. These methods have all taken different choices in approaching the problem. We have different programming languages, different speeds, and so on. But what these 6 that already exist have in common is that they look for cells and nuclei that look like empty barcodes and then remove them. And we were thinking about this and thinking that maybe that would not actually be the best approach, because we don't really know what these empty barcodes are and how that relates to either good or bad cells. So what we set out to do is to try to do something different, which is to look directly for cells and nuclei that look good and then keep them, rather than trying to exclude the bad ones. We look for the good ones and try to keep them. And the way we do this is in an automated way. And we chose to do it fully automated because we had a lot of data sets to process, and we were getting a bit fed up with looking at histograms and box plots to find proper thresholds. And it was also starting to feel a little arbitrary where we were putting thresholds, and it was not really clear how that affected analysis. So we wanted to build this tool that could automate the process for us, to take it out of our hands and to attempt to do it also in a more unbiased way. And what valiDrop does is that it takes a barcode times feature matrix, a raw count matrix, and it needs to be the unfiltered one, so one that can come directly from, for example, Star Solo or from Cell Ranger. And then the first step is to find barcodes that are sequenced enough that they are putative cell or nuclei containing barcodes. And the way we do this in valiDrops is that we use stepwise or piecewise linear regression to find segments of this graph. And then using those segments, we find this breakpoint here where there's a positive angle that we can use to detect this change and find this place in the graph, which is often related to as the ambient plateau. And barcodes associated with sequencing less than this first bend are assumed to contain only free-floating RNA. And we have good evidence that that is the case, because we can see that in poorer samples, this ambient plateau actually moves upwards. So there's at least anecdotal evidence that this level correlates with the amount of ambient RNA. Then what happens after that in valiDrops is that we use a series of community standard metrics to filter barcodes. We look at the relationship between the number of detected features and the number of UMIs. And again, we use piecewise linear regression to fit a curve. And then we find outliers, which is cells that have a unnormal relationship between the number of features and the number of UMIs. We also filter based on the mitochondrial content. Sorry. We filter based on the mitochondrial content. There's evidence in the field that barcodes associated with high mitochondrial content are poor barcodes. What we have seen is that there's a relationship between the mitochondrial content and the number of features detected, and we can use that relationship to analyze the graph and automatically set a threshold. And it's a good idea to automatically set a threshold because different species and also different tissues within species have different levels of expected mitochondrial RNA. For example, humans and mice are different, but also very metabolically active tissues, for example, brown adipose tissues, could be very different or or heart cells could also be very different than more metabolically inactive cells. Then what happens next is that we filter based on the protein coding fraction as well. We have seen that this improves data quality. It doesn't often remove a lot of cells, but it seems to remove cells that are of poor quality. And then the next step, these are the community standard QC metrics that we use. And then the next step is actually based on differential expression analysis. And we do this in a sort of peculiar way. We cluster the cells or barcodes that pass all of these filterings, and we do that at 2 different resolutions. We do it at a very fine-grained resolution, which is the one that's depicted here, where we get a brown, green, blue, orange, and a light gray cluster, and all of the other ones as well. And then we also do it at a much lower resolution, where, for example, we would find only one cluster containing all of these 5 clusters up here. And then what we do is that we test then each of these fine-grained clusters against cells that are not in the same coarse cluster. So, for example, the blue cluster here would not be compared to the orange one because we know that they're related, but it would be compared to all of the other cells down here. And then what we do is that we find clusters that are unspecific, which means that they don't really have any marker genes, or the marker genes are ubiquitously expressed, and we get rid of those. So this is looking for good cells. So essentially, we are looking for cells that have distinct transcription profiles. And then in the last thing that valiDrops can do, which is sort of which is unique at the present time at least, is that we can also look for dead cells and flag dead cells, and that can allow you to analyze them if you're interested in dead cell processes, or also remove them. And I will, in this talk, I will talk a bit about both parts. So the quality filtering path, but also the dead cell removal part. And we have internally, we see this as a few steps in the algorithm. So this point here is stage 1. These community filters are stage 2. The differential expression is stage 3, and this is then stage 4. And once we had built this, we were then left with the struggle of how to actually show that it's good, because there are not really any data sets out there where we have a ground truth, where we know which cells are good and which cells aren't. So we had to come up with our own way of defining what we think is a good cell, and how to validate quality control tools. So to that end, we came up with 2 assumptions, which is that good cells, they map consistently to cell types and states, whereas poor cells assign spuriously to cell types as their profiles are mixed. That means that if a barcode is mostly ambient, then it will be a little random which barcode, which cell type it will look like, because its transcription profile is a mixture of several different profiles. So having these assumptions in hand, what we did is that we ran our quality control metrics or quality control tools, which gave us cells that were putatively good. And then we took a tissue atlas. This is from the islets of Langerhans, for example. And we used Azimut, which is a tool by the Satir lab, to transfer labels from the tissue atlas to the sample of interest. And then we ask several questions. We ask, how certain is Azimut that the label is correct? How consistent are the labels across different label levels? And how well do the labels separate, and so on? So this gives us metrics that we can use to compare our methods to existing methods. And we can also compare internally within our method how well these stages are performing. And that's where we started. So we started with comparing different stages of valiDrops. And what you're seeing here is a peripheral blood mononuclear cells from 10x Genomics, where we have performed stage 1 filtering. And then we have embedded it in UMAP space and transferred labels. And what we see immediately is a lot of these reddish cells, which are erythrocytes, or red blood cells, which are actually not supposed to be in the sample at all. So this indicates to us that these cells are actually poor cells that have high ambient loads. They are strewn out between T cells and monocytes, which is not something you would expect to ever find. And they are also in different places in the UMAP here. They are in this arm as well, going from the B cells toward T cells, and also in this arm here. So they are likely to be poor cells. And what we can see is that we actually specifically remove these cells in the next stage in valiDrops. Now we don't have an arm going between these T cells and monocytes any longer, but we still have residual tails of these poor cells. And then in stage 3, this problem is further limited, and there are no spurious assignments left over here in the T cell area, and a few down here in the monocyte area. And we see really nice separation of subtypes of B cells up here. We have naive B cells, and we have intermediary B cells and memory B cells, and so on. So looking at this visually, it seems that valiDrops throughout these stages is improving data quality. We can then ask, based on these metrics that we have developed, whether we can also see the same thing in a more quantitative way. And if we look at the label scores, perhaps first I should explain how this is set up. So this is data from the same sample, and here we have all of the barcodes which were kept after stage 1, stage 2, and stage 3. And here we have the barcodes that were removed between stage 1 and stage 2, and the barcodes that were removed between stage 2 and stage 3. And what we can see is that Azimuth becomes increasingly certain that the labels that it's assigning is correct with increased filters, with increased stages in valiDrops. And we can also see that the barcodes that are removed are much lower certainty than the ones that we are keeping. We can also look at what we have termed label consistency, and I'll just go back one slide to explain what that is. So these labels come in different levels. So for example, here we have 4 different types of CD4 T cells, but there's also a level in here where they're just called CD4 T cells, all of these 4 groups. So what we did is that we transferred several layers of labels, and then we ask if a cell is labeled as a CD4 T cell on one level, is it then also labeled as a children of that label on the next level? And we can see that that label consistency improves to almost perfect consistency, where labels are transferred only from parent to child, whereas the barcodes that we remove are again much less consistent than the ones that we keep. We also analyzed how separable the labels are in principal component space. We used the silhouette width, or average silhouette width for that, and we also used a metric called LSI, which is the local inverse sampling index, where the silhouette width indicates how tightly clustered together labels are, or cells within a label are, and how much they're separated from other labels, whereas this metric tells you about the neighbors, how often a cell of a particular label has neighbors of the same label. And what we can see again is that filtering with valiDrops improves this massively, and the barcodes that are removed are scattered across the PC space, and we can see the same tendencies over here in the neighborhood analysis. Looking at gene expression, we used entropy as a measure. So here we are measuring the entropy within a cell type label. And if there's a low entropy, then it means that the expression profiles of cells within the same label is high. And we can see that indeed entropy decreases with filtering, suggesting that the labels or the barcodes that are signed to the same labels are increasingly similar, and the barcodes that are removed are widely different from the rest of the cells in the same label. You can see that the profiles correlate really nicely with bulk RNA sequencing data, whereas the barcodes that are removed do not. And we also find an increased fraction of what we call discriminatory marker genes, which are marker genes that can separate cell types from each other. Finally, we also try to cluster the cells and see how well clustering can recover these transferred labels. And we evaluate that using different metrics. Here we have the adjusted RAND index and we have the remeasure as examples. And we can see that the filtering improves the ability to separate these cell types in this data set, whereas the barcodes that we removed are not very nice at separating cell types. So we're quite confident that valiDrops is doing a good job at removing barcodes that are not very informative. So the next question is then to compare valiDrops to alternative methods. And we did that starting with just one data set. Staying in the PBMC world here, we took a data set again from 10x Genomics, which were expected to contain 8,000 barcodes or cells. And here we find that all methods except Dropkick and MTNN find approximately 8,000 barcodes. Our tool is the most stringent one, finding approximately 7,200 cells in this data set. And then we can look at all of the same metrics. We will have valiDrops in red all of the time. And then above each bar, there will be a number which indicates which tool had the highest rank or the best performance for the particular metric. And if we look at the label scores, they're very similar across tools, except maybe over here where MTNN and Dropkick has a bit of a lower performance than the rest of the methods. valiDrops does perform the best across both of these metrics, but it is a close call for label scoring. Label consistency is the same. It's very close. All of the tools are doing a good job, but valiDrops is again the best one. For expression similarity using entropy, we are starting to see more differentiation between the methods. Here again, lower is better because that means that the gene expression profiles within cell types is more similar. And we can see that the tools are starting to separate out into two groups, perhaps for this data set, valiDrops, MTDrops, and SCP2, which is actually based on MTDrops, are doing pretty good. And the other methods are doing quite a lot worse for this particular data set. If we look at separability again, using the silhouette width and LLSI, we also see some differences here. We see that valiDrops has the highest silhouette width, which is the best here, because that means that the labels are tightly clustered and more separated from other labels in principal component space. And we also see that in neighborhood analysis that that cells found by valiDrops are the most well behaved, in that sense that they have most neighbors that are of the same cell type. Finally, we look at clustering, or what we have termed clusterability. So how easy is it to separate the cell types using clustering? And here we're using RI again, and we're using NMI as well, and vMeasure. And we see across all of these metrics that valiDrops is outperforming the other tools. Perhaps MTNN and Dropkick is doing an okay job here, but they were the ones that found the most cells and had really high expression similarity. So it seems that for this particular data set, valiDrops is doing a a much better job than the alternative methods. So what we did from here was that we expanded this test to a lot more data sets. We took it into, I'll just skip that slide, into 47 different data sets from 5 different tissues, 4 different tissues. So we have PBMC, lung, islets, and then we have using single-nucleus RNA sequencing. We also have PBMC and we have brain. And if we look at the individual data sets, there's a lot of information hidden within these numbers. But what we see across all modalities or all data types and tissues is that valiDrops has the best performance across all of them. And the worst performer is also pretty consistent. This method, which we also were struggling to actually get to work. What we did more than just looking at individual data sets is that we also took the data sets after filtering and we tried to integrate them and then look for how much batch effects are there in the integrated space and how able are we to cluster labels in the integrated space. And in these integration tasks, we also see that valiDrops is performing the best, followed up by Cellbender and mtDrops, whereas mtDrops was the second best performer for the individual data sets. And I would say that mtDrops is probably the community standard as it is right now, and it's also doing a pretty good job. We're just slightly outpacing it. We looked into then each individual data set and each individual metric and asked how often is one particular method the best one. And it's shown down here in the fraction. And in red, we have valiDrops again, and we can see that valiDrops is consistently performing good across metrics, and it's the best in approximately 50% of the data sets. And we never found a data set where valiDrops was worse than the third best performer. One thing we did notice, however, was that the expression similarity metric was sort of not great for valiDrops in the integrated space. So we looked a bit closer into that and found that the metrics here are generally very similar. So although valiDrops ranks between 7th and 2nd, it's minuscule differences that actually separate tools, and the same is true for the other metrics that goes into this. So in summary, I would say that quality control tools are hard to benchmark because there's a lack of ground truth. We used label metrics after label transfer as a proxy to do this. And using that, we found that valiDrops sequentially removes low quality barcodes. And compared to existing methods, valiDrops generally achieves better quality filtering. So just moving on to the second part, unless you want to take, I think we will take questions in the end, if that's okay. So in the second part, we're dealing with clinical samples. So one thing we noticed was the clinical samples are often of lower quality because surgery comes first, sampling comes second, as it should. And that means that sometimes sample viability can be a concern. And here's this data from a 10x Genomics technical note where they have a sample with high viability, which they find nice cell types in with distinct marker genes. Then they took the same sample and they artificially decreased its viability by treating it with digitonin. And what they found was that there are, even in a very low viability sample, there's still cells that pass QC, but these cells don't really have any nice cell type markers. So low viability samples can confound good data sets because they do pass QC. There are methods to reduce or remove dead cells. And these methods were tested in a recent paper by Hanamsagar and colleagues, where they did either no cleanup or different types of cleaning up. And what they found was that only column-based cleanup or density-based cleanup reduces the fraction of dead cells. So for example, low spin washes, there's no difference between the relationship between dead and alive cells. There's just fewer cells. But even after column-based cleanup, which is, for example, max filtering, there's still a significant proportion of dead cells left. So what we wanted to do was that we wanted to try to develop a method that can find these dead cells and see if we can remove them. And there are a few data sets out there which deals with this issue. There's a data set from O'Flanagan and Odenes Rueda. In O'Flanagan, what they did is that they took a cell line and they had it untreated, and they used FACS to find live cells and did 10x genomics in those cells. They also took a same cell type, introduced apoptosis or cell death using TNF alpha, and they FACS sorted out dead cells and also did 10x genomics of that. Similarly, these guys, they had hex cells and they introduced apoptosis, but then from the same sample, they separated out live and dead cells. So with these data, we have data sets where we have ground truth because we know live and dead cells. And we use those data to build a classifier for finding dead cells. To build this classifier, we needed to initially find some labels to work with. And what we noticed was that there's evidence in the field that, for example, dead cells, they have higher mitochondrial content than live cells. There's definitely still dead cells that have low mitochondrial content, but they do have higher generally. We also see that they have lower ribosomal content and also lower total reads. And finally, this is a bit hard to, this graph, I think it's a bit hard to read, but on the x-axis here, we have the ribosomal content, and on the y-axis, we have mitochondrial content. And then in the colors, we have sequencing depth. And for live cells, the deeply sequenced cells have high ribosomal and low mitochondrial, whereas for dead cells, it's the opposite. The deeply sequenced ones have high ribosomal and, high mitochondrial and low ribosomal. So it seems that we can use these quality metrics to generate labels. And that's exactly what we did. We used these labels as a weak predictor of being a dead cell. So we have noisy labels based on these metrics. And then what we did is that we took and made a principal component analysis. And we used a setup called adaptive resampling. It's not something we came up with, but it's a method of sampling labels in a way that is amenable to relabeling cells or labels in machine learning. And we built a small classifier using rich regression to actually identify dead cells. And there's a lot more details about this in the paper. Importantly, what we can find is that we can find almost a perfect prediction of dead cells using this on the data sets that we used to create the method, which is of course a caveat, but there's only two. So for example, here we have the truth, the ground truth here, where gray is live and red is dead. And this is the prediction where dead is again red. And we can see that we do recover all of the dead cells using this method. The initial labels here is a weak predictor. Logistic regression improves it a little bit, but our method significantly improves it. And the same is true for the other data set, where we don't find a perfect correlation, but a pretty good one. Subsampling analysis shows that our method is pretty sensitive. We can find down to approximately 3 dead cells with pretty good confidence. And we also looked at when the MCC is low, which is when we have a low accuracy, is that because we mislabel dead cells, or is it because we mislabel live cells? And what we find is that we only ever mislabel dead cells. So sometimes our method will misclassify a dead cell as live, but we will never misclassify live cells as dead. So this means that you will not actually lose anything by using this method, because we don't have these false positives. So we set out to test this, and I will skip this in the name of time. And the way we did this is that we analyzed a data set from Madsen et al., where they had stored lung, esophagus, and spleen samples. They processed them fresh and after storage. And what they found using tunnel staining was that the lung has an mildly increased, or well, not mildly increased, has a mild level of apoptosis, and it's not changing a lot between 0 and 72 hours of storage, whereas the spleen and the esophagus has an increase in apoptosis. And what we could see is that our tool can recover this. So here we have the fraction of dead cells, and we can see in light green we have fresh, and after 72 hours we have, or in dark green we have 72 hours. And we can see in the spleen and the esophagus, our method predicts an increase in the number of dead cells, consistent with the tunnel staining. We then wanted to ask if we can then rescue these 72-hour samples by removing the dead cells. And what we could see is that the dead cells are not uniformly distributed across cell types. So it seems that some cell types are more prone to dying in these storage conditions. But importantly, what we could see is that that the cells that are predicted to be live are much more similar to the fresh cells than the cells that are predicted to be dead across these cell types. So we can almost recover the original transcriptome if we remove the cells that are flagged as dead ones, looking at pseudobulk. So in summary, dead cells can pass quality control, and they can also confound single cell and single nucleus RNA-seq datasets. Our methods can, with high accuracy, detect and flag these cells. This enables either their removal, if that's your interest, or also their analysis, which can also be of biological interest. And importantly, we do so with very low false positive rates. So there's not really a lot to lose, because we don't mistakenly remove live cells. So to end, I would just like to thank a few people, my group, and especially here Gabia in red here, who has been one of the main drivers of this work, and of course collaborators at the Novo Nordisk Foundation Center for Genomic Mechanisms of Disease, and also in Atlas and funders. And finally, I would like to thank you all for listening and take any questions.
Right, great. Thank you so much for the great talk. It's very interesting talk. Well, please just, if you have question, please just raise your hand or just type your question in the Q&A. I might have one question before. Oh, I saw that there's two more questions. I have one question. Like when you try to use the ASMERS, you know, the reference basic database, single cell RNA-seq database to transfer labels. I was just wondering, like, if we have a tissues that we don't have any reference database we can use, how should we do that? How should we actually use your tool?
So my tool is not dependent on having a tissue database. We just only used it for validation purposes. So we only, to validate our method, we only chose samples that had a reference atlas, just to validate it. But okay. It can be used for any tissue from any sample.
So basically, if we don't have any reference database for my tissue, for example, here, we just cannot do the validation step, but we can still use the tool to filter out noisy cells, right?
Yes.
Okay, great. All right, we have one question from Biju. Let me just unmute you. Okay, so Biju, I think you can talk now.
Hi, can you hear me?
Yes.
Hi, thanks. That was an interesting talk. I had a question about in your first part, you had summarized that if you have expression similarity, and something that you have mentioned in your paper too, where if you were to work with a cell line, a pure cell line, the valiDrops has issues with dealing with that data set. Is that like a general, or if there is expression differences in the cells within the sample from a cell line, the valiDrops will still work, or generally the cell?
Yes, that's a good question. So because valiDrops is using these differential expression analysis to find good barcodes, it doesn't work if there's no heterogeneity in your sample. But if there are populations or subpopulations that you suspect should be different, then it will work. So a clonal cell line, I don't particularly know why people would do single cell RNA sequencing on clonal cells, but if you do, then you shouldn't use valiDrops. But if you have a mixture of cells or treated cells or anything like that, then it should still work.
Okay, great. Thank you.
All right, Daniel, you can unmute yourself.
Thank you very much for the great talk. I have a question about identifying the empty droplets in your knee plot analysis. I know that you use localized linear regression. Have you guys compared how it performs with the approach used by CellRanger?
So I think CellRanger is, or at least was, using the knee point detection method of empty drops, and that finds a more stringent point on the graph than we do. So we haven't actually compared it directly to CellRanger because the later CellRanger versions uses empty drops.
Yeah, so to use valiDrops, we should use the raw matrix returned by CellRanger, right?
Yes, not the filtered one, the raw one, yeah.
Yeah, that's great. And another question is about how reliable is the labeling of the dead cells in single nuclei RNA-seq? Because in that approach, we deplete the cytoplasm, and then we get rid of the mitochondria. So the formula strictly has hardly code the proportion of the mitochondrial content in there. Is it transferable to be used in single nuclei RNA-seq, or is just to single cell RNA-seq data?
Yes, that's a good question. The prediction of dead cells is only possible in single cell RNA sequencing currently. And that's exactly because we use mitochondrial content to softly label the cells initially, and you cannot do that in single nucleus RNA sequencing. So it doesn't work.
Okay, thank you so much.
Welcome.
All right, we have one more question from Ashish. Ashish, I think you can unmute yourself now.
Hey, hi. Can you guys hear me?
Yeah.
Yes.
Okay, yeah, thank you. Like my question is on the part of the tool. So you actually mentioned in your slides, there are 3 steps we can do the QC on. So can we just do the first 2 steps or the first step for our pipeline, or there is a way to do that in the tool?
Yes, there is a way to do that. So all of the functions are available individually, and then there's a wrapper function that goes around it. Okay. So when we in my group process, for example, smart-seq data, we only use the first 2 steps of the method, and not the last one. Oh, well, actually not the first one. So we only use stage 2 for non-droplet based data.
Okay, thank you. Yeah.
All right, great. I don't think we have more questions, but I think you guys feel free to explore this tool and get feedback to Jesper. With that, I think thank you, everyone. Thank you, Jesper, again, and for a nice talk, and have a nice day.
Thank you.
Bye.
Bye.
Bye.